Myself, Professor Sujata Mahapatra from the Department of Botany, Rama Devi University, Bhubaneswar. In this video lecture, I shall be discussing about the basics of a stir tank bioreactor. It is a part of course curriculum in BSc Botany, then MSc Life Sciences, MSc Industrial Microbiology, and MSc Biotechnology course. These are the contents of this video lecture: introduction, standard criteria for designing the bioreactor, types of bioreactors, components of basic stir tank bioreactor, application of bioreactors, and conclusion. Bioreactor and fermenters. These are two equipments which are used for research purpose or industrial production of certain products. They have several applications and they are industrially very significant instruments. Bioreactor and the fermenter, the difference is fermenters are used mostly for the production of microbial biomass and microbial products, and bioreactor is used for the culturing of any type of living cell, may it be animal cell, plant cell, or a microbial cell. And there is a process that is bioprocessing. It is a technique that is for the production of the industrial pharmaceutical enzymes and proteins from living cells, and bioprocessing is done inside a bioreactor or fermenter. These are the different applications of the bioreactor. It is used for the production of nutraceuticals, pharmaceuticals, methanol, ethanol, single cell protein, primary metabolites, secondary metabolites, and food processing. These are the different types of bioreactors. So based on how the bioreactor gets operated, there are different varieties of bioreactors. Based on the structure and operation procedure, there are different types of bioreactor: continuous stir tank bioreactor, bubble column bioreactor, airlift bioreactor, then fluidized bed bioreactor, packed bed bioreactor, and photobioreactor. Stir tank bioreactor. It is a type of bioreactor in which stirring is done. It is fitted with a stirrer, and inside a bioreactor, generally there are three phases: solid, liquid, and gas, and there is a proper mixing of these three phases inside the bioreactor. For this proper mixing of the phases in a stir tank bioreactor, there is a motor driven shaft fixed with the agitators or impellers, and this rotates, and when it rotates, all the three phases get mixed up. There are different types of stir tank bioreactor: microbial bioreactor, cell culture bioreactor, and a single use bioreactor. Continuous stir tank bioreactor, as opposed to the stir tank bioreactor, is a open system where there is continuous addition of nutrient and removal of the equal amount of the spent medium, microbial waste products from the vessel. In a stir tank bioreactor, usually batch fermentation is done. So once the nutrient is added, and at the end of the process only the product is recovered. One meaning of fermentation is it is an anaerobic pathway in which glucose is converted to simpler compound with the release of energy. A more appropriate and practical definition is it is either an aerobic or anaerobic process during which chemical changes occur in organic substances through the action of enzymes. Fermenters are used for microbial fermentation, and bioreactors are used for any type of living cell culture and bioprocessing. Configuration and design of a fermenter or the bioreactor. Many things are considered while designing a bioreactor because the product recovery, the quality of the product, and the quantity of the product depends upon the design of the fermenter. Inside a fermenter, many chemical reactions occur and microbial biomass or cell culture is done at an optimal condition, at optimal conditions. Due to this, the design is very important, and there are many things are taken into consideration like fluid dynamics, biochemical changes, pressure development, etc. One important aspect of the construction of the bioreactor is the material which is used for the body of the bioreactor. It may be glass, it may be stainless steel, or it may be wooden. For the production of alcohol and lactic acid, wooden fermenters may be used. But mostly in the industry, stainless steel fermenters are used because of their durability, and stainless steel may be mixed with some amount of nickel and chromium to produce a stronger and durable body of the bioreactor. Second important consideration is the aseptic condition or maintenance of sterile condition during the operation of the bioreactor. For certain products like the antibiotics, very stringent sterile condition is required. These are high cost and low volume products. And physical and chemical sterilizing agents are used. Proper sterilization is done before fermentation and after the fermentation also. The inlets and outlets which are at different places of the fermenter are properly sealed to avoid contamination. So different types of sealing are used like the O-ring sealing, gasket sealing or leaf sealing is done. Microbial fermentation systems or any bioprocessing system has three phases: solid, liquid and gas. So there are three types of reaction: liquid gas, liquid solid and gas solid reactions. Proper mixing of different phases and mass transfer of liquid and homogeneous distribution of all the contents is required to achieve maximum product and for the proper maintenance of the optimal condition. This is done with the help of a stirrer which is fixed with the blades of the impeller. This is also called as agitator, and when it rotates, the contents are homogeneously mixed. Head space and aspect ratio. Inside a fermenter, there are two areas. One is working volume, the other is the head space. Working volume is the area in which the actual process occurs, the microbial culture or the cell culture happens, and above that place, above that space, there is an area that is called as head space to accommodate the foam and to accommodate the gases for their release like carbon dioxide. And there is a proper ratio is maintained. Head space, the standard working to head space volume is 70 to 30 in a basic bioreactor. There is a manhole at the top through which a man can enter and clean the interior after and before the process, and sterilant can be used properly. So there is inoculation point also for the aseptic transfer of the inoculum. There is sampling point to take out sample from time to time and check their quality and different other parameters. Expenditure analysis is very important because it is an industrial process and there should be reliable operation. There are different types of operation depending on the condition of the addition of the inoculum, addition of the medium and withdrawal of the products. There are three types of operation. First is the batch fermentation. Batch fermentation is a closed system, means there is no exchange of matter. From the beginning, all the things required like the microorganism or the cells or the inoculum and the nutrients are added, and after adding everything, proper sealing is done, and at the end of a duration, particular specific duration, only the fermenter is closed, fermenter is stopped, and the product is recovered. In between, only oxygen, antifoam agents, or the acid base for pH adjustment is done, but nutrient is added from the beginning and product is recovered at the end. No in between taking out of the product or addition of nutrient in such kind of fermentation. Fed batch fermentation. It is also a closed system like the batch process. The only difference is in this type of operation, substrates are added in small batches. At time gaps, small amount of substrates are added because sometimes, as in case of the production of antibiotics like penicillin, heavy amount of substrate from the beginning represses the synthesis of the antibiotic. Mostly for the production of secondary metabolites, fed batch fermentation process is used because the heavy amount of substrate will result in catabolites repression or the repression of the synthesis of mostly the secondary metabolites. Third type is continuous fermentation, and the difference between batch and the continuous fermentation is it is an open system. There is exchange of both matter and energy. Sterile nutrient solution is continuously added to the fermenter. Product containing the medium and microbial mass is simultaneously removed from the fermenter. There are two types of continuous fermentation operation. One is homogeneously mixed bioreactor and the second is plug flow reactor. This is a cross section of a stir tank, continuous stir tank bioreactor. There are different parts. This is the sketch diagram of a stir tank bioreactor. The construction is very important. Configuration is important. Head space working volume is also important. So before the designing the bioreactor, care is taken and many calculations are made to create a particular dimension of the bioreactor, and the base is curved. Curved base is very important. Then the shape of the bioreactor is almost rectangular. The jacket is thick. The body is thick to withstand pressure because heavy amount of pressure is generated. The wall is made up of stainless steel or glass. Then this is the shaft carrying the layers of impellers. Impellers are the blades which are fixed to a central disc. All the discs are carried by the shaft. The shaft is attached to a motor, and when the motor rotates, the whole thing revolves and mixes the content homogeneously. These are the baffles. To all the walls, usually 4 baffles are added, and if the size is larger, more baffles are required. Baffles are metal plates attached to the walls of the fermenter. During the agitation, vortex is produced, and the vortex may create problem in the mass transfer process. Therefore, these baffles are fixed to dissolve the vortex to prevent the vortex formation. These are the baffles. Then there is a pH monitor, pH controller, which will record the pH change from, and it will show what is the particular pH at a particular time, and accordingly The pH can be maintained by the addition of acid or base through a pump. Acid or base is stored in a reservoir. Reservoir is fixed with a pump, and the pump will automatically add the required amount of acid or alkali from time to time to maintain optimal pH inside the fermenter. This is the medium. This is the culture broth or the medium. Then these are the bubbles, air bubbles. Air bubbles are required or the air bubbles of the oxygen is required in aerobic fermentation, and sparger is the device which puts the air bubble into the medium, which adds the air bubble to the medium. The smaller the bubble, larger is the surface area, and it is better for the operation of the bioprocessing event. Smaller bubbles are important, but more pressure is required to pass these bubbles through these small holes. So that there are different types of sparger, but the spargers are provided with holes, and from bottom the sterile air or oxygen comes and passes through these holes, and it is added to the medium, and that is the sparger. Then there is, during this operation, heavy heat is generated. There are different heat dissipating devices. One is shown here, that is the cooling jacket through which water circulates, cold water circulates throughout the outer wall of the fermenter and keeps the interior cool. Then there is sampling sterile seal through which the shaft is introduced. It is sealed properly. There is viewing port. There is steam injection to maintain temperature. Then also for sterilization purpose. Then there is sampling port, viewing port. So these are the main parts of a stir tank bioreactor. Gas distribution is very important for the proper mixing of the gas. There are different types of devices like stirrers, pumps, or overpressure of the gas. For the maintenance of the aerobic living cells, oxygen is important. And oxygen, before entering into the unit of the fermenter, is sterilized. Sterilization of the oxygen is achieved by using different filter devices. Different types of filter like the single membrane filter or pleated membrane filters can be used. Membranes made up of Teflon material are more durable. Sparger is another device that adds the small air bubbles into the medium during bioprocessing. These are the different types of sparger, and choice depends upon the operator or the objective of the operation. So these are so many types of sparger are available. Impellers. Impellers are the main parts of a stir tank bioreactor. Impellers or agitators, based on the direction of fluid flow, are of three types. Axial impellers. Fluid flows along the axis of the shaft, and it is mostly used for certain purpose. Then radial flow impellers. Fluid flows at right angles from the shaft radially, and mixed flow impeller. Fluid flows diagonally. Impeller means there are blades which are fixed to a shaft, and number of impellers depends upon the working volume. The higher the working volume, more number of impellers are fixed to the shaft. The shaft is attached with a mechanical device, that is motor, and when the motor is on, the shaft rotates, and the impellers mix the contents. Two types of bioreactor like bubble column and air-lift fermenter have no requirement for agitators because there the mixing is done by some other technique. These are different types of impellers. So this is the shaft, this is the disc. To the disc, these blades are attached, and this shaft is attached with a motor. When the motor is on, the shaft rotates, and these impeller blades rotate in one direction and they mix the contents. So there are different types of impeller blades. Again, choice is done basing on the what type of products is required. Then baffles. Baffles are blades attached to the wall of the fermenter. During fermentation, vortex is formed. Vortex, it creates a area of separation of the gas from the other areas. So here there is trapped gas, and that hampers the bioprocessing event. So to dissolve the vortex, baffles are fixed to the lateral walls of the fermenter, and when the baffles they rotate to and fro, they will dissolve the vortex and mix it to a homogeneous condition. So baffles are meant to prevent vortex formation. They increase the turbulence and aeration, and overall they improve the mixing. Then there is acid-base reservoir, and the acid-base reservoir is fitted with a peristaltic pump, and that adds acid or base to adjust the pH. There is a pH monitor that records the pH, and automatically the peristaltic pump acts and there is addition of required amount of acid or base. So the pH, this is the diagram of a pH probe that records the pH, change in pH, and automatically pH is adjusted. Neutralizing agents are used, but care should be taken that they should not affect the living organism or the internal environment of the bioreactor. Temperature control is very important because inside the bioreactor, the conditions must be maintained at optimum. The temperature, pH, and the concentration of substrate must be maintained properly. Heat is generated during the microbial activity and due to the agitation, mechanical agitation also. And heat exchanger, there are different types of heat exchangers that redistribute the heat and maintain the temperature. Some vessels are equipped with a cooling jacket, and cooling jacket is made up of a flexible material with holes. Through the holes, the coolant flows, and there are different types of cooling devices in different types of fermenter. And there is a particular device called an exit gas cooler that removes moisture from the gas exiting from the fermenter. Then there is sampling point through which sample can be withdrawn from time to time to check different parameters and quality of the product. Sampling point is used, but after use again it is sealed to prevent contamination. Foam breaker, foam is accumulated during fermentation. It comes from the substrate or the products. And if the foam mixes with the contents of the fermenter, it may produce low quality product. So foam has to be dissolved. There are anti-foaming agents that are used, or there are like the vegetable oils can be used, or mechanical foam breaker can be used. So that was a discussion on the basics of the star tank bioreactor. It has several applications, very important applications. Mostly now it is used for the research purpose, for the in cell-based therapy industry and tissue engineering. These are the medical applications of the bioreactor, and new bioreactor are fixed with modern technical features. Some examples of the modern bioreactor are microfluidic bioreactor, 3D bioprinting bioreactor, etc. So bioreactors were first produced during the 20th century, and in the past years, it has undergone tremendous change in the design. Now it is fitted with computer control condition, and it is a high-tech device. In the future, it will undergo still more evolution because its applications are increasing day by day. Thank you.